Hello, good morning to everyone. We'll wait a couple of minutes to start. Thanks for joining us. It's great to have you today. My name is Andrea Diez. I am the marketing leader at Path Group, and we are very pleased to have your presence. We hope that this session will provide you with useful insights and innovative ideas that you could apply later on your business. Before to start, please let me inform you that this webinar is being recorded and then we will share it with you. Microphones and cameras will be shut down to avoid interruptions, but please, if you have any questions or comments during the presentation, you can write into the chat and we will respond them as soon as we finish. If some of the questions are not answered due to the time, you will receive the response by email shortly after. Additionally, I will write down my email address, and if you have any comment or suggestion, you can reach me. Thanks for your patience. We will wait a couple of more minutes, maybe 2 more minutes, until everyone can join us. For introducing our speaker, let me tell you briefly about Path Group. We are a leading firm of over 35 years of experience in the pharmaceutical, nutraceutical, food, cosmetic, and veterinary industries. As you can see in this video, our comprehensive approach combines research and development, laboratory services, international validations, advanced industrial machinery, and technical training, partnering with clients from the initial idea to the final product. With operations and innovation laboratories in Spain, Argentina, and the United States, we deliver solutions and specialized consulting to clients in over 50 countries. Now, let me introduce you today's speaker, Mr. Gabriel Porto. He is PhD pharmacist. He is director at Path Group with large experience in research and development, regulatory, quality, and production pharma processes. Let's welcome Gabriel, who will be sharing his expertise and insights on how innovation is transforming nutraceutical processes from laboratory development to real market applications. Thanks, Gabriel. Good morning.
Good morning, everybody. Thank you for the introduction. Welcome, everybody, to join us. So we hope that you can have a good webinar together, and hope to see most of you in next fair in Barcelona Vita Foods. Okay, so we'll start. So today we are going to talk about our experience on nutraceuticals. So we'll talk about how to go from lab to market in many applications that we have in nutraceuticals. So our tablet of content is today will be to talk about the market overview in the European region, to talk about production challenges that we normally have, the technology and how we can apply. The case studies that we are going to talk about are probiotics and digestive enzymes and functional infusions, and we'll close with some conclusions. So, concerning European nutraceutical market, we can make a summary saying that there are in the recent years growing is very strong. The key drivers are that really a rising health and wellness awareness, an increasing prevalence of chronic diseases, Asian population seeking preventive healthcare. So it's taking a lot of impulse. So consumer trends are also uh, something that is pushing it. The innovation, advances in ingredients technology and product development that allow to make new formulations and new technologies. The market enablers, in fact, the regulatory support and expansion of the line distribution channels that we have in our industry. So making like a kind of market share of pharmaceuticals going to nutraceuticals as well. And the outlook, sustained growth ahead with opportunities for manufacturers, CMOs, suppliers, and investors. Okay. So talking about the market size, it's growing very, very fast. We estimate to grow from 92.83 million this year to more than 117 million in 2031, with a compound annual growth of almost 5% in this period, in the forecasted period. Okay, so as you can see in the summary there, it's considering rising very, very fast. And we consider it's a market that is gaining some areas to pharmaceuticals market. So talking about opportunities and challenges, in fact. So first, talking about the market opportunities, we can say that it's rising demand for natural and organic compounds. Consumers increasingly prefer plant-based, clean label solutions, so less artificial, let's say. The preventive healthcare awareness, so growing adoption of dietary supplements and functional foods. The expanding Asian population, consistent demand for products targeting osteoporosis, cognitive health, and mobility, mainly. The technological innovation, with advances in ingredients extraction and formulation that enables more effective, viable, and differentiated nutraceuticals. And the rapid advancement of personalized nutrition fueled by artificial intelligence, wearable health tech, and microbiome testing, creating research in demand for tailored supplements based on individual biomarkers and lifestyle data. So, what challenges do we have in this market? Well, we have a varying and strict regulations across European countries, increasing compliance cost and delayed product launches that, of course, with the time that will be more strict. So at the very beginning it's maybe soft and then it's coming more hard. Then the market skepticism. Consumers have some doubts about the scientific backing and support, and if it's something safe. So that's makes some doubts in the consumers. Then the supply chain disruptions, sometimes dependence on raw materials from diverse regions, exposes the market to geopolitical and logistical risk. You cannot find same, I know, kind of herbals or roots or some, something like this available everywhere and with the same specifications and so on. Then the intense competition, the proliferation of brands that needs continuous innovation and marketing efforts to maintain the competitive advantage. No? And then finally, the competition from conventional pharmaceuticals, as we said before, is like kind of market competition, market share with the nutraceuticals and pharmaceutical products. No? Then, so nutraceutical face this competition from conventional pharmaceuticals that are backed by extensive research, regulatory approvals, and some established marketing channels. So there is a competition over there, right? Well, let's talk a little bit about the production challenges that we afford. So we have here a table that summarizes some examples, okay, of technical challenges, the impact on production they have, and solution, potential solution. So normally we have a kind of challenge like biomass variability that we can have an impact on production of different potencies and quality in the different batches. So maybe fermentation equipment and process control within light sensors that are able to control stability and have reproducible parameters is the solution, right? So we have processing costs that usually have an impact on the higher energy consumption in drying and concentration. So by using efficient drying technologies like optimized spray drying, fluid bed dryer, reduce costs and maximize the yield. Then some other challenges we front are product stability. In fact, we have some example today, case study on this point, this issue. So the impact on production is the loss of biological activity in prebiotics and structs. And the solution is encapsulation and granulation technologies with humidity and temperature control systems to prevent or to preserve the functionality of the prebiotics or these actives. Okay. Then we have others like regulatory compliance, so which with an impact on the requirement for traceability and validation. So to use GMP documentation and regulatory support protocols, validations and training is necessary. So powder physical properties are some other challenges that we have in this field, with a poor friability, composite, dosing problems, uniformity content. So normally to use granulation and mixing equipment that improve homogeneity and flow properties are the solution. This is also something that we are going to have as an example today. And then, for example, competitive differentiation with saturated market, difficult to stand out. So to have the opportunity of R&D and pilot to industrial scale-up services, enable launches of innovative formulations with technological backing, it's really a solution for these issues. So this is just an introduction of different production challenges that we have to every day to manage. Okay, so let's do some approaches on how we can manage these challenges that we have daily. So typically, we based on technology and we based on formulation and know-how to solve these challenges. So through technology, by product differentiation, with advanced technologies that enable companies to create unique formulations and line extensions of existing products. No? That so by using technology, you can manage and you can have this freedom to manage this opportunity. Uh, and of course, by formulation and know-how, we can make nutraceuticals more effective, safer, and differentiated. Mm. So this by using the know-how, you can bypass inconvenience of compression, inconvenience of flowability, inconvenience of this kind of problems that usually we have to to manage and to challenge daily. Mm. So what about, let's talk about a little bit so about technology and how. Which tools do we have there, right? So let's say that the technology are critical for stability and performance. To what we want to say with that we can manage and we can improve our possibilities by using technology and formulation know-how. Okay, so here we have an example of different technologies on the left and the principal advantages and limitations do we have with each of the technologies. Okay, so here we are comparing different technologies to be used. First, on the left, it's mentioned the high shear mixer plus fluid bed dryer, where the principle is that the high shear mixer you granulate particles and then spend in hot air with controlled flow in the fluid bed dryer. Okay. Advantages. Okay, it's fast granulation and homogeneous drying. Have an excellent control of moisture and particle size. It's suitable for heat-sensitive products with short processing times because the granulation is very short and the drying is also quite short. Allows granulation and coating processes. Limitations that requires particles of adequate initial size and not always appropriate. You will see, in fact, today one of the case studies showing that it was not as good as the top spray solution. So the second one we are mentioning there is the top spray, the fluid bed dryer. So the principle is atomization from above onto the fluid bed, combines drying and granulation, okay, at the same time, improves flowability and compressibility of product, encapsulates its actives to protect them from degradation is something possible to do with the top spray. It has very high reproducibility. So what the limitations are? That may require fine parameters adjustment to avoid excessive agglomeration. So it's very important the experience and the adjustment of parameters during this process to avoid problems of agglomeration and sticking. But then we have the traditional spray drying, which is the most, maybe one of the most knowledge, where you have the principle of atomization of a liquid into a chamber with hot air. It's really fast and scalable. It's very well known in the industry, as we said. But you always have a VAT. You have a higher risk of thermal degradation, less control of particle size, powders often less dense with durability issues. So it's really, really very difficult to manage. And something that is not mentioned maybe here is that the cost is very, very high. Also, when you have a spray drain process compared with the previous ones, okay, it's more expensive. And finally, in the table, we have levelization, freeze drying, where the principle is the freezing and sublimation of water. The advantages we have that preserves biological activity very well because it has no impact on that. It's ideal for sensitive proteins and prebiotics. The limitations are that very high costs. The processes are very long processing times, and the powders are very fragile, difficult to handle in compression or encapsulation. So you get a very fine powder, and it's very fragile. Uh, so it's a very expensive technology and has this inconvenience. So it's applied only in selected opportunities. So let's talk about 5 key reasons why technology is key. Um, First, stability of the active ingredient. Many nutraceuticals contain sensitive compounds like prebiotics, enzymes, plant extracts that need proper drying prevents thermal degradation, loss of biological activity, and contamination. So we see in the case study an application in this sense. Then viability and clinical efficacy, the physical form of the powder, like a particle size, density, residual moisture, influence on how the body absorbs the nutraceutical. Then scalability and industrial efficiency is also very important to have a fast and homogeneous drawing process to reduce the production times, energy cost, and also to avoid stressing the product inside during the process, right? Then the regulatory compliance, the FDA and the different regulatory agencies is also very important. Traceability and validation of the processes. So technology applied should be good enough to have these possibilities. And the versatility in dosage forms. The drawing determines whether an ingredient can be transformed into capsules, tablets, sachet, or soluble powders. So advanced technologies allow the properties such as flowability, compressibility to adjust the dose. Okay. So let's go now to to show you the case studies that we perform for some of our customers in our lab. Some challenges that we have to pass through and to find the solution. Okay. So the first case study we talk about is prebiotics and digestive enzymes. In fact, to this field, this case it was a prebiotic, and we will show you the case. So the challenge was how to from liquid biomass to gaseous form, right? So transform the biomass into a dosable powder. It's not just a technical step, it's a bridge that converts science into real solution, we said, because we see that seems so easy, but in fact it's not. So in this process has strategy values because we can ensure stability and quality. So with the powder preserves the bioactive compounds and extends shelf life. Facilitates precise dosing. Uniform powder ensures accurate ingredients amount per dose. This is very important because we are in nutraceuticals and the doses are also very important. It's not a food, it's a nutraceutical. So allows for industrial scalability, powder format facilitates transport, storage, and mass production, opens the door to innovation that's versatile, integrates bioactives into diverse products, different forms, and builds consumer confidence. No, consistency guarantees durable benefits and credibility. So, as you can see, this is a real picture of the product we have to manage. You can see the biomass, and our chance was to go from this liquid biomass to powder, to a dosage form, powdered, feasible to be uh, transformed in a capsule or tablet or sachet. so, so in this case, uh, we start with a liquid biomass. That can be a microbial culture, plant extract. In this case was uh, this was the case, was a prebiotic. Then it's converted into stable powder through process such as fluid bed and top spray. That was the technology we used. The powder is dosed into dosage forms like capsules, tablets, sachets, as I said. This process allows bioactive ingredients to be scaled up, stabilized, and commercialized. So it's a way to, to commercialize it finally. You know? So we started with biomass production, with fermentation, with controlled cultivation of the microorganism. It's alive. It's probiotic. Then we have a concentration, separation and concentration by centrifugation, filtration, and extraction. Then we started with this biomass, with the controlled drying in the fluid bed dryer, where we use top spray technology to transfer the biomass into stable powder. That was the key point, where we passed through from liquid to powder in this process at our lab. And then with this powder, this stable powder that we will show you then after how it was, we got a stable powder feasible to be encapsulated and that and put it in a matrix like a capsule. Okay. Then after you have downstream, you have the powder homogenization with mixing with different ingredients or excipients, and the quality control and final dosing in capsules, sachets, tablets, or functional beverages. Okay. So, uh, so which was the challenge? The client provides a liquid biomass, like cream, containing the Saccharomyces cerevisiae. This is an active microorganism. The objective was to evaluate the feasibility of different production processes and technological considerations, how we are going to confront this, in order to determine the suitability to obtain a product that meets quality standards for dosage administration while preserving microorganism viability and stability. That was the great challenge. No? So knowing that this temperature and other factors are very critical. So we have the Saccharomyces there. So the main challenges that we identified were preserving microorganism viability during and after production process, obtaining a solid form suitable for incorporation into different product matrices for the dosage administration, incorporating organoleptic characteristics, aroma, taste, sensory experience without affecting product integrity, and ensuring product stability over time, so the shelf life. Okay. Um, so the trials were conducted using different manufacturing processes and experimental approaches designed to evaluate process robustness and its impact on microorganism viability. That was the main concern that we had during this development. So critical parameters, process temperature, the microorganism residence time, that because as the longest, the worst, we should make it as short as possible. And the medium acidity was also a concern. So what we did was to test two different methodologies. One was the fluid bed granulation using top spray system, the first one. And the second method was to use the high shear mixer granulation followed by fluid bed drying. So as you can see in the pictures, we use only one equipment, but by using top spray or high shear mixer plus fluid bed dry. In both cases, we use the same raw materials, of course, and same conditions to compare the results. So we saw significant differences between both methodologies. In method one, with the top spray system, we have higher process efficiency, better impregnation of the creamy phase onto the solid substrate, as we use a substrate to receive the liquid cream. Preservation of microorganism was okay. Improved sensory properties of the product were okay, and uniformity particle size distribution was acceptable. You see a real picture of the product over there. In the method two, we use high shear mixer followed by fluid bed drying. What we observed was lower operational efficiency and higher equipment requirements. So it's really more expensive as we had to use two equipments. Failure to meet microorganism viability criteria, we couldn't maintain it, as well as in the method one. And heterogeneous particle size distribution with fines and large agglomerates distributed in the powder, as you can see in the picture. So the final results, the final results met specifications using the method one, obtaining a product with physical and biological characteristics aligned with the requirements quality standards. Activation and variable count analysis were performed to compare pre and post, so that we saw the results that we maintain the potency of the Saccharomyces after the process. And additionally, final sensory analysis of the tasting confirmed an overall improvement in product experience. So this translates into reduced production cost, greater efficiency in raw materials use, maintenance of product quality, lower equipment investment, and expanded portfolio and final products. So Well, this was a video that we performed summarizing what we have obtained. Okay, let's go to the second case we're talking about this morning. It was about functional infusions. Okay, as we said, totally different application, totally different. Here, just to introduce a little bit in this field, in the tea industry, this is we are now to the experience we did was with the herbals in the tea industry. We said that every detail matters. So normally the the consumers are very strict, no, on what they receive. So we said that the structure of the leaves, their stability, and the proper incorporation of sense are critical factors to guarantee a high quality product. And however, when these elements are not properly controlled, problems arises that directly impact the final results. So considering this that the consumers are very strict, we must have a good compromise on the finished product. So in this case, we start from the raw materials that are the loose leaf tea in processing, the micro ingredients, and the liquid flavorings. So the three that were already as raw materials, uh, the liftees were processing and conditioning in this in a controlled drying and selection of plant materials. The micro ingredients also were stabilized as a micro ingredients to be added to this leaf tea, and we had to add liquid flavorings in a controlled form, okay, to ensure homogeneous distribution and final product stability. So we have the stage of blending with the liquid injection and atomization. So what we use through our BBC, the IBC blender technology, uh, essence and liquids can be directly incorporated into the blend, ensuring homogeneous distribution and precise process control. You know that typically we use in our industry, in fact, IBC are not typically adding liquids by injection, atomization. So that was our challenge, in fact, to use the BBC IBC blender technology by adding a liquid in this way. And you will see in the case study how it's compared by using atomization compared with no atomization. So finally, we had the powder homogenization, mixing and particles control to ensure uniformity, and final product was a tea sachet. Okay. So the challenge was that the client used the mixing method for tea blends that showed opportunities for improvement in flavor incorporation and in achieving a more homogeneous strand structure. So the process objective was to maintain the structure of the leaves without breaking the leaves, okay? The stability and the proper incorporation of scents to guarantee a high quality product. So we faced two main issues: breakage of tea leaves due to their high fragility, very fragile, and the formation of clumped product affecting blend homogeneity. So additionally, there was a need to incorporate scents that would enhance organoleptic properties, aroma, flavor, sensory experience, without compromising the product integrity, so without breaking the tea leaves. So at our lab, we applied two technologies and we compared them. First, method one was control incorporation of liquids during the mixing process with the atomization, as we said. And the second one was the addition of the liquid at the end of the mixing cycle, followed by remixing. So this was the method used by the client until then, where the liquid was not atomized. So the trials were carried out under the same conditions with same inputs, same proportion, same composition, of course. What we got, the results were very clear. In the method one, we have greater efficiency in processing times, more controlled, improved impregnation and more uniform liquid distribution, preservation of leaf structure, so we have no breakage of them, and enhancement of sensory properties. In the method two, although it was delivered the expected results since it was the method used by the client until there, we have lower intensity in aroma and flavor and slight degradation of final quality. Okay. The agglomeration was and the not uniform distribution was the main issue there. Okay. As we will see. So the results exceeded expectation. We have intact leaves, no breakage, homogeneous blend, greater aroma and flavor intensity, better distribution and uniformity, with improved product texture. So let's see the final results. We have so the development revealed something very key, that was that the new method intensified the pel's properties by using, so you use less aroma to obtain the same results. And we have finally that translates into reduction of production costs, greater efficiency in the use of inputs, improved product quality, and a 25% increase in production efficiency and sensory performance. Let's see how it performed. I have some images, some video with the same.
Andy, can we please check the audio? Because we are not hearing.
Oh, you cannot hear it?
Not yet.
No. Wait. Strange. I will do it again. Sorry for that. Okay, I will try again. Can you see my screen? No.
Yet.
Okay. Can you see the presentation again now?
Oh.
No? Oh,
Oh.
okay. Let me see what happened. I have no issue. I am sharing it. You can also only see me, right? Not the presentation.
Correct. Sorry for the inconvenience.
Okay, I will go out and in again. Sorry.
Sorry for the inconvenience. We will be back shortly. Otherwise, I will share the presentation. We will see a nice video. It's a pity that we cannot hear that, so we are trying to fix. In the meantime, please remember that you can write your comments or questions in the chat, and we will try to respond them during the webinar. Otherwise, we will respond by email shortly. I will write down my mail in case you want to leave a question later on.
No, it's okay, right?
Yeah, that's okay. Now we can
Okay.
see the
Sorry for that.
thing. No
Okay,
problem.
let's go to the video.
In the tea industry, every detail matters. Leaf structure, stability, and essence incorporation define product quality. When not controlled, performance and final results are affected. The challenge. The client faced two issues: fragile leaves breaking, clumping that reduced blend uniformity. Additionally, essences had to enhance aroma and flavor without damaging product integrity. The challenge: keep strands intact and achieve uniform textures and flavors. The solution. Our team tested two methods in its pilot lab. Method 1, controlled liquid incorporation during mixing. Method 2, liquid added at the end, then remixed, client's current method. Same inputs and conditions were used. Method 1, faster, better impregnation, uniform distribution, intact leaves, improved sensory properties. Method 2, expected results but weaker aroma/flavor and slight quality loss. Positive lab results were scaled and validated in production. The results team expertise was key to success. Method 1, efficient, preserved leaf integrity, uniform particles, enhanced sensory profile. Method 2, lower aroma/flavor intensity, slight degradation. Outcome, the new method intensified properties, reduced need for essences, and saved time and cost. This case shows that success is not just mixing, but understanding process and product behavior. Pat Group develops applied knowledge that delivers real results.
Well, it was okay, right?
Yeah, perfect.
Okay.
Thank you.
Welcome. So, in conclusion, we have solutions for producing nutraceuticals, prebiotics, and vitamin complexes, ensuring efficiency and quality at every stage. So, here we are showing you some real experiences we did have with these different customers, where we had to work on prebiotics, capsules, or granular powders for reconstitutions, as the one we showed, transforming from liquid biomass to granular powder. We use the top spray process, and we allow that extended shelf life of the microorganisms by using this technology. We apply that for Saccharomyces, as we said, Enterococcus, Lactobacillus, among others that we work with. We also did that for digestive enzymes like phytase, amylase, among others, using again top spray process for extended shelf life of the product stability by using the same, practically same technology and same application. Then we also worked, for example, for multivitamins and minerals with a chewable, effervescents, or coated tablets and powders for oral reconstitution, like B12, calcium, magnesium, okay, different zinc, remember lecithin, L-cysteine, I don't remember what else, but with different vitamins, vitamin C. Okay, by direct mixing processes and granulation, we use technology that are applicable to this combination of minerals, vitamins, and amino acids. And then again, the other one what we presented today, the functional infusion with the herbal strands and powders. So we showed the blending of herbals, strands and powder with the liquid injection inside the BBC, the IBC. That's something totally different that you can find in the compare with you can find in the market as a solution. So it was very successful. Another like granulative masking for animal protein, also for powder for constitution and mixing into food. It was something that we did like masking of taste and odor. Is also application. Normally you don't use high shear mixer plus fluid bed dryer. Sometimes top spray is also a possibility, but typically works better in high shear mixer and fluid bed dryer. Okay, so thank you very much for joining us today. We
have one question. Maybe we can take these few minutes before we finish and answer to Anna. Anna is asking, she says, it's interesting how fluid bed dryer was used for liquid products. Was it a standard machine, same as used for granulation, but without powders, or optimized mechanically for spray drying?
No, it's a fluid bed dryer. It's the same machine. The fluid bed dryer is the same machine, but we use a top spray drying. It's not spray drying. It's top spray. So it's the same machine, basically same machine where we use a gun. So we use a nozzle to apply the liquid into a powder. So in fact, technology, what we use is the follow. Normally, when you are drying a powder, you enter to this machine, to the fluid bed dryer, with a wetted granulate, okay, powder, and you dry it. In this case, what we put inside is like a powder, yes, not wetted, a powder inside the machine, inside the fluid bed dryer, and we add to it the liquid. While adding the liquid, we are evaporating the solvent. So in the liquid is the biomass with is a solid in a liquid. So we what we are doing is to put the solid with the liquid to another solid. Yes, inside the fluid bed dryer. That's why the technology is very interesting because you use the same machine as you use for drying, but in this case for top spray application. I will stop this.
Perfect. Thank you, Gabby. Well, we want to thank everyone for joining us today. We will respond the questions or your concerns or any ideas that you would like to share with us by email. Remember that we will be present at Vita Foods in Spain, and we hope to see you there. We can book a meeting and and meet each other. Thank you. Have a nice day and see you soon. Thank you.
Thank you, everybody.
Bye-bye.
See you. Have a good day. Bye-bye.